Today we will start a new chapter, hormone. So you are already acquainted with the term hormone. There are different hormone secreting glands which are called, these are the specialized tissue usually known to us as endocrine glands. But there are some cells which secretes hormone but not organized as glands. Okay. So today we will see what is the definition of hormone, what is the classification. Classification is very important because depending on the type of hormone, depending on the classes of hormone, its mode of action differs. Okay. So general mechanism of hormone we will discuss. Once you know what is the general mechanism of hormone, then you do not have to go the individual mechanism of hormone. Depending on the different endocrine glands, different type of hormones are secreted. You will know what gland secretes what type of hormone and what is the mechanism of their action, what is the biochemical function of the hormone, of those hormones, and what is the deficiency disorders created by excess function of that hormone or deficient function of that hormone. That we will discuss in the subsequent classes. Now we will take a concept of general mechanism of hormone action. Now definition. Here are the 4 points. These 4 points you have to write in definition. You should not miss any of the 4 points. These are the hormones are the organic molecules, organic substance produced and secreted by ductless glands in minute quantities that regulate various physiological and metabolic function. So in definition, these 4 points you have to mention: organic substance produced in minute quantities, produced and secreted by the ductless glands, which regulate various physiological and metabolic functions. Okay? Now, E.H. Sterling, he introduced the hormone, this term hormone first time, and Bayliss and Sterling discovered the first hormone secreting. Now, one thing I would like you to remember, in the definition of in the internal assessment we asked you vitamin D function as hormone, justified. Okay, many of you have written that is synthesized in the cell, carried to the effector organ by blood. Okay, now here I have not mentioned in the definition, it is carried by blood to the target organ, because there are three types of hormones. You know, some hormone, they are synthesized by the cell and released in the extracellular fluid and acts on the cell which secreted it. So you can clearly see this hormone is not carried by blood. It is secreted by the cell. It is produced in the cell, secreted by exostosis to the extracellular fluid. Once it is secreted in the extracellular fluid, it acts on that cell which secreted it. So there is not carrying system here. That's why I intentionally omitted that part, that it is secreted and carried by the blood, but it is there. So such type of hormone we call as autocrine hormone. Autocrine hormone, auto means cell. So the cell which secreting it So, the cell is producing it. What is the need of secreting it? Because the receptor for this hormone is located in the cell surface. The receptor of the hormone is located in the cell surface. So, once the hormone it is produced, it will be released by exostosis in the extracellular fluid. Then it comes here and bind here in the receptor. That is the need of secreting the hormone in the extracellular fluid. So this type of hormone which act on the cell itself, which is producing the hormone, is called autocrine hormone. This is autocrine hormone. Now another type is there. It is secreted by the cell in the extracellular fluid, but do not bind to the cell secreting it. Rather, in the adjacent cell it binds. Okay? Adjacent cell is the target cell. So it binds here. Okay? Now it is also not carried by the blood. So auto means acting on the cell, secreting it. Para means acting in the cell adjacent to the cell secreting the hormone. Okay? Para means side by side. So paracrine cell, the hormone secreting cell is releasing the hormone in the extracellular fluid that binds to the adjacent cell. That type of hormone is called paracrine hormone. Example is nitric oxide. It is synthesized in endothelial cell. It diffuses not by blood. It diffuses to the smooth muscle of the vasculature, and there causes relaxation of the smooth muscle, thereby dilating the vessel, facilitating the blood flow. So example of this is nitric oxide, and example of autocrine cell is your IL-2. IL-2. It is interleukin 2. Now, example of autocrine hormone is IL-2. It is secreted by the T lymphocyte, released in the extracellular fluid. It binds to the T lymphocyte receptor located in the cell membrane of the T lymphocyte and causes proliferation of T lymphocyte. In case of nitric oxide, it produced in the endothelial cell, diffuses to the smooth muscles of the vasculature and causes vasodilation. Now the last type of hormone, this is endocrine hormone. They are secreted in the endocrine glands. They are released in the extracellular fluid, taken up by the blood vessels, carried to the target cell. Here you can say that, in definition, you can say that it is produced in the Daclus gland, carried by the blood to the target organ and exerts its function there. Maximum of the hormones are endocrine type of hormones. Endocrine type of hormone. That's why if you say that it is transported by blood, your definition will not be wrong. So all other type of hormones, they are the endocrine type of hormones. So 3 types of hormone: autocrine hormones, paracrine hormones, and endocrine hormones. Okay. Now classification of hormone, it is very important, because depending on the classification, you are going to see what is the mechanism of its action. It is classified depending on the structure, function, and mode of action. Based on the solubility, they are classified into 2: lipid-soluble, water-soluble. Lipid-soluble or lipophilic, and water-soluble, they are hydrophilic or lipophobic. Significance of this classification is that those which are lipophilic in nature, they can cross the membrane bilayer easily and do not require any receptor in the cell surface. They can enter the cytoplasm through plasma membrane without any assistance. Example is nitric oxide. Nitric oxide can diffuse inside the cell. So lipid soluble hormones, example is steroid hormones, mineralocorticoids, glucocorticoids, androgens, estrogens, progestagens. These type of hormone secreted by which gland, you will come in the next class. So we will confine today in this only. Just know it: mineralocorticoids, glucocorticoids, androgens, estrogens, progesterone, etc. Then also thyroid hormones, nitric oxide. These are all lipophilic hormones. Water-soluble hormones, they are the catecholamines, epinephrine, norepinephrine, peptide hormones, then protein hormones, okay, like insulin, glucagon, thyroid tropin releasing hormone, thyroid stimulating hormone, parathyroid hormone, oxytocin, vasopressin, calcitonin, gastrin, etc. They all are water soluble and they cannot cross membrane bilayer easily without any assistance. That assistance is provided by a transmembrane integral protein called receptor. Transmembrane integral protein called receptor. It binds to the receptor as like of your enzyme binds with substrate. Similar mechanism. Okay? It binds with the receptor and exerts its function. Now, it binds to the receptor. So, it is the first messenger. Hormone is the first messenger in case of group 2 hormone. Hormone that are lipid soluble are called group 1 hormone, and hormones that are hydrophilic are called group 2 hormone. Okay? Depending on the second messenger they generate, again group 2 is divided into group 2A, group 2B, group 2C, group 2D, and in some book they have written group 2E also. Now, based on the chemical nature, classification based on the chemical nature, amino acid derivatives, you know, catecholamines, thyroid hormones, okay, nitric oxide generated from arginine, this is from tyrosine, okay, melatonin from tryptophan, steroid hormones derived from the cholesterol and has steroid nucleus. Steroid hormone, as it is derived from the cholesterol, it will have a steroid ring in its structure. So, they are this is the examples: peptide or protein hormones and fatty acid derivatives. Fatty acid derivatives, one fatty acid is there, arachidonic acid. From that, some hormones are generated like prostaglandins, leukotrienes. So they are the fatty acid derivative of fatty acid derived hormone. Classification of hormone based on mechanism of action. As I told, group 1 hormone, these are lipophilic hormone, can cross plasma membrane without any receptor, without any assistance. Okay, they to exert this function, they also need receptor, but that receptor is located in the inside the cell. So that is called intracellular receptor. Now intracellular receptor, it may be in the cytosol or it may be in the nucleus. Okay, so intracellular receptor may be cytosolic intracellular receptor and or nuclear receptor. Group 2 hormones that bind to the plasma membrane receptor, they are usually hydrophobic. They are usually hydrophilic, and here hormone is the first messenger and its action is mediated by generation of second messenger. So once it binds to the first cell membrane receptor, it cannot produce its action. It has to generate, it has to undergo a series of signal transmission so that second messenger is generated. So hormone that bind to the plasma membrane receptor, and hormone is the first messenger for this group. Action is mediated by the second messenger. Depending on the second messenger, they are group 1A, group 1B, group 1C, group 1D, and in some group 1E. In those groups which have not mentioned group 1E, this calcium is incorporated in group 1C. Okay. Up to this much, do you have any problem? If you understand these things, you can add up from the book, and I think you need not add also more than this. For your MBBS curriculum, this is more than enough. Mechanism of action of hormones. Here are the different hormones, how they generate second messenger. Let's see in the next slide how it is. Now, group 1 hormone. This is the hormone. This is the plasma bilayer. So this is the plasma bilayer. In plasma bilayer, the hormone, as it is lipophilic, it can easily travel through the hydrophobic core of the plasma membrane and comes into the cytoplasm. Here it unites with the receptor. This coupling of hormone with receptor is called hormone receptor complex. Okay. Now cytosolic receptor, it is a mobile receptor, it can move. Okay. Now hormone receptor complex, as it is attached, as it is mobile, it can cross through the nuclear pore to the inside the nucleus. Okay. Now in nucleus, they have a specific area of binding site in the DNA. That site is called steroid sensitive hormone responsive element. Let's see in the next slide. Here. So here are the hormones. They enter in the cell cytosol. Okay. There is intracellular receptor is there, cytosolic receptor. Then receptor and steroid hormone complex is formed. It crosses the nuclear membrane through nucleopore and binds in the DNA strand. Receptor, steroid hormone complex and DNA interaction is generated here. Okay. Now, in the receptor, in the DNA, it binds to the hormone responsive element. This hormone responsive element is located in the upstream of the promoter region. Say, this is a DNA. Okay. Now here is the gene. This is 5' end. This is 3' end. This is the proximal end. This is the distal end. Okay. Now, before this is a gene. Before gene, there is a area called transcription initiation site. Okay. Now, before transcription initiation site, there is another region called promoter region. This part is called downstream, this part is called upstream. Okay. Now, upstream to the promoter region is the hormone responsive element, this part. This is the hormone responsive element. That receptor and hormone binds here. This hormone responsive element is usually steroid sensitive area that can recognize the particular specific steroid hormone. That's why in this hormone responsive element, that particular hormone only, hormone receptor complex only can bind. So after binding, the promoter region start functioning and the gene becomes activated. So there is transcription. Remember, there is transcription. Transcription of mRNA. After a few days, you will start having class on genetics. That there you will find what is mRNA, how this transcription is done. Okay, then it will be clear for you. Just for the time being, you remember that transcription of mRNA is done. Now this mRNA, here is the mRNA Now this mRNA, it will come out of the nucleus through the nucleopore to the cytoplasm. Okay? In cytoplasm, there is translation. Remember the terminology of specific protein. So in cytoplasm there is transcription of specific protein encoded by that gene. Okay. Now this protein is responsible for different physiological and metabolic function. One example is calcitonin, calcitriol. That is acts via this group 1 mechanism. It causes synthesis of cal binding protein in intestinal cell that helps calcium absorption. Okay. So this is the protein which is responsible for metabolic and physiological response. This is the mechanism of action of first group of hormone. Okay. So far we have known definition of hormone. These are, can you tell me 4 points? Anyone? 4 points in definition? Anybody? These are organic substances produced in minute quantities. Okay. Then third point, produced and secreted by, tell me, fourth point. Okay. Now they are of 3 types: autocrine, okay, autocrine, paracrine and endocrine. Maximum of the hormones are endocrine in nature. Okay. Now they are classified depending on the structure, function and mechanism of action. Okay. So here we are discussing the lipophilic hormones, their mechanism of action. Lipophilic hormones, they can cross plasma membrane easily without any receptor. Okay. They enter in the cytoplasm, combine with the cytoplasmic receptor which is mobile in nature. Hormone receptor complex enter in the nucleus, bind in the hormone responsive element and trigger transcription of mRNA from the responsible gene. Okay. Now this mRNA, it comes out of the cell, out of the nucleus, and in cytoplasm the required protein is translated. Okay. Required protein is synthesized. The term is translation. So let's go to the second group. So this is the group 2 hormone, not group 1. Group 2 hormones. Usually they are hydrophilic. They cannot cross plasma membrane easily, needs assistance of receptor. Receptors are located on the cell surface. So, hormone that binds to plasma membrane receptor, there is activation of G protein, activation of effector protein, and there is generation of second messenger. Now, how it happens? Cell surface receptor. We will know a few words about cell surface receptor. You may get a short note receptor. When you get this as receptor, then you have to discuss cell surface receptor and cytosolic receptor and nuclear receptor. Okay. Now, whatever you had known in the cytosolic receptor, you just mentioned that a nuclear receptor is the hormone responsive element. That hormone crosses to the nuclear membrane and binds directly with that hormone responsive element. That is that function as nuclear receptor. Okay. Now, major is your this one cell surface receptor. These are integral membrane protein, binds hormone like substance binding in enzyme, 3 functional domain, extracellular, transmembrane. Now, this is an integral protein. Okay. This is the integral protein. This will have extracellular domain, then your transmembrane domain and intracellular domain. Extracellular domain is responsible for hormone binding or hormone binding. Transmembrane domain, hormone it cannot cross the membrane. So, transmembrane domain, it will undergo conformational change along with this one, intracellular domain. Okay. Now, first messenger is bound to the receptor, sending the signal to the intracellular domain. In intracellular domain, in the intracellular domain, it remains coupled with a specialized type of protein called G protein. Okay. Now, G protein, you may get a short note. Okay. G protein, it is a intracellular membrane protein having 3 subunits bound together. Alpha subunit, beta subunit, gamma subunit. In alpha subunit, it remains bound with GDP, guanosine diphosphate. The gamma subunit and beta subunit and gamma subunit, they remains tightly attached. Whenever they dissociate, these dissociate together. Okay? And alpha subunit, it remains loosely bound with the beta and gamma. So, when this alpha subunit, it gets betas very easily. Now, binding of the hormone. Binding of the hormone to the receptor causes conformational change in the intracellular part of the receptor. That causes conformational change in the alpha subunit of the G protein. This is the G protein. These three subunits are all together, they form a protein called G protein. Now, receptor coupled with G protein is called G protein coupled receptor. Okay. Now, once the hormone is bound, the alpha subunit, there is a conformational change. With the change of the conformational status of the alpha subunit, there is less affinity of the alpha subunit to GDP. So, when there is less affinity, what it does? The GDP is changed into GTP. Okay? GDP is changed into GTP. GDP is changed into GTP, replaced rather, GTP. So, alpha subunit become alpha GTP. This part you have to mention only if you get short note on G protein. So, GTP, and is alpha bound, GTP bound alpha protein is an active form. Now, this active alpha GTP complex, it stimulates the effector protein. E is effector protein. So, you can see there was a binding of first messenger, that is hormone, in the receptor, and it has transmitted so many signals for generating second messenger. Up to now, there is signal transmission only. Okay, there still the second messenger is not generated. Okay, now it is the effector protein which is responsible for generating the second messenger. So, you understood this much? You understood this much? Hormone binding in the receptor causes conformational change in the intracellular domain of the receptor. That in turn causes conformational change in the alpha receptor. The affinity of alpha receptor for GTP is reduced and replaced by GTP. Once it is bound to GTP, it gets dissociated from the beta and gamma subunit. It becomes separated, beta and gamma subunit gets separated, and active form of alpha GTP subunit, it stimulates activity of the effector protein. Now, what is that effector protein? Let us see. Now, hormone receptor interaction transmits a signal across the membrane to protein complex called G protein. Now, G protein bound G protein through G protein coupled receptor, it transmits signal through the G protein for the generation of second messenger. This process is called signal transduction, sending the signal to generate second messenger. In short, this is the diagrammatic representation. Cell surface receptor. Now, 3 subunit of GTP, alpha is bound to the GDP. Okay, it is in resting state. Now, the hormone bound to this, there is replacement of GDP. GDP is moved out and GTP is attached with the alpha subunit, and alpha subunit gets activated. G protein dissociates into beta, gamma and alpha GTP. Alpha GTP is the active form. Alpha GTP stimulates the effector protein. Okay. Now, what are the effector proteins? I will tell you later. Once it activates the effector protein, it has to get inactivated. Otherwise, you will go on producing the second messenger and there is devastating situation in the body. Okay, the action will be more than the required. So, it has to be inactivated. How it is inactivated? This alpha subunit, it has an intrinsic this action of GTPase action. GTPase action is intrinsic GTPase activity. So, after stimulating the effector cell by the action of intrinsic GTPase activity, this GTP is converted into GDP. Okay? Converted into GDP. You can see here inorganic phosphate is removed and GTP is converted into GDP. GDP bound alpha protein is inactive and it has an affinity to bind with the beta and gamma subunit. So, once it becomes alpha bound GTP, it gets attached with beta and gamma subunit and becomes ready for the second turn of action. You understood now? You have tried to understand. I think this is the easiest way I described it. So, binding of cell surface receptor stimulates alpha GDP to get converted into alpha GTP, gets dissociate from the beta and gamma subunit, traverse to the effector protein to stimulate it. As soon as it stimulates the effector protein, it gets inactivated by the GTPase innate GTPase action of the alpha subunit into alpha GDP. Alpha GDP is inactive form and has affinity towards binding for gamma and beta and gamma subunit. So, it gets bound and it becomes ready for the second fresh action. Now, this is the mechanism by which the effector protein is activated. Now, about the G protein. Signal transducing protein, primer located in the cytosolic part of the membrane. It has 3 subunits alpha, beta, gamma. Alpha is remains bound with GDP during resting state. Hormone receptor coupled means G protein coupled receptor gets activated and activation of G protein occurs. There is conformational change in the G protein. GDP is replaced by GTP in alpha subunit. Dissociation from beta and gamma occurs. Then alpha GTP active form stimulates effector protein. Now, these effector proteins are adenosine, adenylate cyclase, guanylate cyclase, phospholipase C. These are the effector protein. Now, what is the function of adenylate cyclase? Adenylate cyclase, it causes ATP into cyclic AMP. Adenylate cyclase two molecular pyrophosphate is removed, it becomes adenosine monophosphate but in cyclic form. Say here is adenosine, adenine, then ribose. Ribose has 5 carbon. With the 5th carbon, say if I diagrammatically represent like that, this is the 5th carbon, this is the 3rd carbon. Okay. Now with 5th carbon, there will be 3 phosphate bound together. Okay. Now adenylate cyclase, what it does? It cleaves here, remove pyrophosphate. Okay. pyrophosphate. Now one side is empty for the phosphate group. P, pyrophosphate is removed. It forms a bond with the 3' carbon. Okay, that's why it is called cyclic AMP. Cyclic AMP. In plain AMP, it is not an active form. There is cyclization is not there. Only phosphate is attached with that. But when there is a cyclization, it becomes active form. This is called cyclic AMP. Now, likewise, in guanylate cyclase is produces cyclic GMP. Phospholipase C, it cleaves inositol bisphosphate into inositol triphosphate and diacyl glycerol. They also act as second messenger. So, after this effector protein is activated, alpha GTP is inactivated rapidly by GTPase activity of the alpha to alpha GDP and gets dissociated from the effector protein and associated with beta and gamma. Second messenger cyclic AMP, ATP by adenylate cyclase converted into cyclic AMP. Now this cyclic AMP acts as second messenger and responsible for phosphorylation of protein kinase A. That will cause phosphorylation of phosphorylation of different proteins, enzymes, which will give this biochemical functions and structural functions. Okay. Stimulation of protein kinase A by cyclic AMP will cause phosphorylation of various intercellular enzymes and proteins. That is the mechanism of action. In case of cyclic GMP, now just few words about protein kinase A. It is called due to its dependence on cyclic AMP. It is a tetramer having two regulatory subunit and two catalytic subunit. Binds with four molecule of cyclic AMP and release catalytic subunit free for phosphorylation of different protein like glycogen synthase, then glycogen phosphorylase. This cyclic AMP, as it is second messenger, needs to be inactivated soon, and it is done immediately by the phosphodiesterase. Now here it's not that clear. This is the regulatory subunit, catalytic subunit. 4 units of cyclic AMP attached with this, and this catalytic subunit is dissociated, gets dissociated and activated. Okay. Now it is responsible for phosphorylation of required protein for biological function. Second messenger GMP, like that of your cyclic AMP only difference is in place of cyclic AMP it will be cyclic GMP, converts GTP to cyclic GMP. The effector protein is guanylate cyclase. It causes phosphoprotein kinase. In case of cyclic AMP it was A, in case of cyclic GMP it is protein kinase G. Phosphorylation of varieties of protein, okay, that cause smooth muscle relaxation and vasodilation. Example, you know, nitric oxide. Nitric oxide, what happens? This is the atrial natriuretic factor. Atrial natriuretic factor. This is another type of hormone, and this is nitric oxide. Okay. Now ANF binds with the membrane bound receptor and nitric oxide binds with the cytosolic receptor. Here is the guanylate cytoplasmic guanylate cyclase. It is this guanylate cyclase acts due to the influence of nitric oxide. So it converts GTP to cyclic GMP. But in case of natriuretic factor, it acts through the membrane bound guanylate cyclase. Okay. So membrane bound guanylate cyclase, it is converts GTP to GMP, cyclic GMP. So both of them acts through cyclic GMP. They generate second messenger as cyclic GMP, but their action or functioning of receptor are different. One function in the cytoplasmic receptor, another functions in the membrane bound receptor. So this is the mechanism how second messenger GMP is generated. So second messenger, when you write in the mechanism, this detailed part you don't have to write. Up to effector protein generation, then mention the effector proteins, cyclic adenylate cyclase, guanylate cyclase for phospholipase C, and adenylate cyclase, it generates the second messenger cyclic AMP, guanylate cyclase second messenger cyclic GMP. Okay, and phospholipase C, it generates second messenger inositol triphosphate, diacyl glycerol and calcium. Okay. Now phospholipase C is activated by hormone receptor complex through G protein. Phosphoinositol bisphosphate is cleaved into diacyl glycerol and inositol triphosphate. Diacyl glycerol stimulates protein kinase C, and inositol triphosphate induces release of stored calcium from endoplasmic reticulum and calcium channel. Opening calcium channel. Raised intracellular calcium act as messenger. One example is calcium calmodulin activity. Stimulates several intracellular enzymes like phosphorylase, kinase, myosin light kinase. These are the examples. Now, in some book, they have mentioned calcium as separate messenger. In that case, the binding of hormone to the calcium ATPase transport system in the cell membrane causes inhibition of the transport of calcium from intracellular space to extracellular space. So this is the transport system. Binding of this to this receptor, not receptor, this is a carrier protein. In case of calcium, where calcium itself functions as second messenger, it binds, it stimulates the protein, activates the protein, and the protein binds with the calcium ATPase channel and inhibit calcium transport from intracellular space to extracellular space. So there is a increase intracellular calcium. So this rise of calcium, it is responsible for influencing the different and activity of different enzymes and producing the biochemical function. Sorry. The fourth one is tyrosine kinase system. One example is insulin. when insulin binds to the receptor or hormone binds to the receptor, there is conformational change in the intracellular domain of the receptor. That conformational change causes phosphorylation, autophosphorylation of the intracellular domain at tyrosine residue of the side chain. Okay. Phosphorylation of the tyrosine residue of the side chain means it is in the receptor. Receptor is phosphorylated. Now this phosphorylation causes increases affinity to a cytosolic protein, specific protein. In case of insulin, it is insulin substrate insulin receptor substrate 1. So, insulin receptor substrate 1 gets again phosphorylated by the activated receptor. Okay? And after that, this into insulin receptor substrate, it goes on series of phosphorylation reaction, cascade of phosphorylation reaction, and mediates biochemical function. Okay? So, binding of cytoplasm protein IRS and phosphorylation of this is followed by series of phosphorylation cascade and brings biochemical function. So, these are the different mechanism of action of hormones. Now, here I told this is cytosolic calcium. This is in some book, it is written in the group C. Okay. Here is the calcium independently, it increases the activity of the protein, like your calcium calmodulin complex they form. Example is glycogen phosphorylase. It has alpha, beta, gamma and delta subunit. Delta subunit is nothing but the calmodulin. So, it binds with 4 molecule of calcium, okay, and there is conformational change and change in activity of the enzyme, causes change in the activity of the enzyme. So, this much detail you may not go, for understanding purpose how it causes, this has been described. So, your main part is by how this second messenger is generated, and the second messenger is responsible for causing the biological action. Okay. So, this may be inhibitory type, this may be excitatory type depending on the type of hormone. Okay. So, this class we will finish up here. Thank you.